Welcome to Anatomy and Physiology's Chapter 10. And with this chapter, yet again, we are just hopping from organ system to organ system. And with this chapter, we're in the muscular system. And like always, before we even get started, you already know some things about this system already. Way back from Chapter 1, remember this system is made up of muscles, and muscles help us to move. And you even know a little bit more about this chapter already because when we talk about muscles, like any other organ system, tissues come back. And so we're talking about tissues as well. But no worries, you know a lot about the tissues. It's the same three tissues we saw before when we looked at tissues. It's skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. And so as we go through this chapter and this organ system, like usual, we're going to look at this organ system microscopically, which is what we're going to do in chapter 10. And then we're going to look at it macroscopically, looking at the organs in chapter 11. So we're going to start off first with the tissues. And again, you know a lot of this already. It's the same three tissues we saw before. It's skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. Again, just think back to when we did tissues. You know already, for example, when we looked at skeletal muscle tissues under the microscope, it had those alternating light and dark bands. Remember, we call those striations. So we say skeletal muscle tissue is striated. It has those alternating light and dark bands. What else? Think back. We saw this tissue. When we looked at each cell, a single cell could have more than one nucleus. And you remember the nuclei looked like they were pushed to the edges or the periphery of the cell. So we say skeletal muscle tissue has multiple peripheral nuclei. Or we say it's multinucleated, meaning it has multiple nuclei. And where did we find this tissue? Remember, it told us in the name. It's called skeletal muscle tissue because it's found in skeletal muscles, the organs, which are muscles we use to move. And because we use them to move, we control them. You remember when you control a muscle tissue, we say it's voluntary. So you can see, you knew all this from tissues already. None of that was new. You already knew skeletal muscle tissue was striated. It had multiple peripheral nuclei. It's located in skeletal muscles, which are attached to your skeleton to help you to move. And you control it. It's voluntary. So let's remind ourselves about the other two types of muscle tissue, like cardiac muscle tissue. Remember, it actually tells you in the name where to find it. Remember, it's called cardiac for cardia, which is heart. Remember, you find cardiac muscle tissue located in the heart. And the heart's there to help pump blood around your body, to help send oxygen and nutrients. And remember, do you have to tell your heart when to beat? Oh, I hope not. Why? Because you don't control this tissue. It's involuntary. But remember, it did look a little bit like skeletal muscle tissue. How? Remember, when you looked at cardiac muscle tissue under the microscope, it also had those alternating light and dark bands. It also had striations. But every once in a while, we saw a really dark-looking band, almost looked like a dark striation. Remember, those weren't striations. Those individual dark-looking bands we saw scattered about were intercalated discs. Remember, intercalated discs are just part gap junction. They're allowing cells to communicate via electricity and send nutrients, primarily to help make sure all the cells beat at once. Remember, you want your heart and all the cells in the heart to contract at the same time. So they're using all the gap junctions located in the intercalated discs to help send that signal. But again, keep it simple. Cardiac muscle tissue, striated. Remember, when you look at the cells in cardiac muscle tissue, they only have one nucleus, sometimes two, but stick to one if I were to ask. Remember, it's located in the heart, and you do not control it. It's involuntary. And then the last type of muscle tissue is smooth muscle tissue. Remember, it's called smooth because when you looked at it, it did not have those alternating light and dark bands. It did not have striations. It was non-striated, meaning there were no striations. And remember, where do you find this tissue? Well, you find it in places like hollow organs. Think your intestines or your blood vessels. They will have some smooth muscle inside to help with movement. And remember, think intestines. You don't have to tell your food when to move around inside your intestines. Why? Because you don't control smooth muscle tissue either. It's also involuntary, just like cardiac muscle tissue. So as we go through this microscopic part of the chapter, we're just really focusing on the tissues here. And to keep things simple, from here on out, as we progress, we're only talking about skeletal muscle tissue. Because you saw from our previous chart and what we already know about our tissues, they are not the same. So if you were to look at each individual type of cell up close, a skeletal muscle cell, a cardiac muscle cell, a smooth muscle cell, they will not actually look the same. So to help keep everyone from getting a little confused and overwhelmed, we're just going to focus on one for this chapter. We're going to focus on skeletal muscle tissue. So from here on out, as you go through the rest of this chapter and this lecture, just stick in your mind that you're only talking about skeletal muscle tissue. And we'll use this because in chapter 11, we're going to be looking at skeletal muscles, the organs. So we're going to look at the tissues first. And again, tissues are just one level of your whole body. Remember, you have multiple levels to you: a chemical level, a cellular level, tissue, organ, etc. And so we're going to attack this at really two major levels: the cellular, or really three: cellular, tissue, and muscle level. So you could imagine, what would I want or need if I wanted to make a muscle cell, or a skeletal muscle organ? Well, it could begin with the cell. Remember, you have a cellular level. So if I wanted to build a muscle, all I really need is a lot of muscle cells. And that's what you see on this chart here, or this picture here. To build a muscle, it begins first with a single muscle cell. And by the way, in case you're using different textbooks, there are lots of names for a muscle cell. You could call it a muscle cell, or a myocyte, MYOCYTE, or a muscle fiber. These are all synonyms: muscle cell, myocyte, or muscle fiber. But looking at this chart, look all the way towards the bottom. We're looking at one individual muscle cell kind of pulled out in the image. And all you're doing to make the muscle is gathering lots of cells. But you got to think, you got to hold these cells together. And the thing that's going to hold these cells together is connective tissue. So as we talk about how you're using this cell or cells to build a muscle, we got to talk about all the connective tissue that's holding and covering all these structures and keeping them in place. I want you to know the three major connective tissue coverings that we find in skeletal muscle. I'll mention their names first and then go one by one like usual. The three connective tissue coverings are endomysium, perimysium, and epimysium. Those are the three connective tissue coverings. I want you to know the three coverings, and this is simply, what are they covering? So let's build this muscle. So if I want to build a muscle, again, I'm going to get my muscle cell, aka my muscle fiber, which is what they call it in this picture. And then I'm going to surround that individual cell actually with connective tissue. This is not the same thing as the plasma membrane. This is going to be connective tissue sitting on top or outside, more superficial to the plasma membrane. And it's called endomysium. Endomysium is the connective tissue that covers a single muscle cell. Keep it that simple. But again, we got to build our muscle. I need more than one cell. So you're going to grab a collection of cells. A collection of cells in anatomy is what we call a fascicle. It's a bundle of cells. In this case, a bundle of muscle cells is called a fascicle. And you're going to have to keep this fascicle together. You're going to have to keep these cells together. So again, we're going to need some more connective tissue. And there's going to be connective tissue wrapped around the fascicle, surrounding this collection of cells, and we call it perimysium. Again, like usual, keep it simple. Perimysium is the connective tissue covering for a fascicle, or a collection of muscle cells. And then to build the entire muscle organ, well, you're going to get a lot of these fascicles together. You're going to get a lot of these bundles of muscle cells. And you're going to need to keep all these fascicles together. You're going to need to keep the entire muscle organ together. And like usual, you're going to use your connective tissue covering to do it again. So the last bit is epimysium. It's going to be surrounding the entire muscle to hold everything together. So that's a little bit on how you make a muscle. There's a bit more involved, but basically, if you think about it of the cells in the muscle, it's just how do you hold them together? And it's going to be these connective tissue coverings helping to do it. One more time, it's going to be endomysium surrounding a single muscle cell, perimysium, which surrounds a fascicle or a collection of muscle cells, and epimysium, which is going to surround the entire muscle. And that's how you're going to build it up from these individual cells. But again, in this chapter, we're not going to focus on muscles, the organs. We'll do that in chapter 11. In this chapter, we're going to focus on the cells themselves. And again, remember, everything we're talking about right now is just skeletal muscle. So where do skeletal muscle cells come from? Well, like any other cell in your body, they start off as very immature cells. We call them myoblasts. And when you look at a myoblast, individual immature cells, muscle cells, actually only have one nucleus. And it's the way they get multiple nuclei is that these cells actually fuse together as they mature into a bigger muscle cell. So whenever you look at a mature skeletal muscle cell with all its peripheral nuclei, you're really seeing the nuclei of every individual immature cell before they fuse together. Like usual, stick to the exam review. And so we're going to look at a skeletal muscle cell up close. We're going to look at a myocyte up close, a muscle fiber up close. Remember, those are all synonyms. And like usual, you know stuff already. It's just a cell. And like all cells, it has those three basic parts to it. It has a plasma membrane, that outer boundary, holding in everything inside, aka the cytoplasm, and it has at least one nucleus. We know it's skeletal muscle tissue or skeletal muscle cells. They have more than one nuclei. It's just that as we talk about different parts of this cell, the names are going to change to help be more specific. For example, I mentioned your skeletal muscle cell has a plasma membrane, but its name changes to help us be specific. Remember, our anatomists want you to know specifically what they're talking about. So instead of saying, hey class, let's look at the plasma membrane of a skeletal muscle cell, I could just say, let's look at the sarcolemma. The sarcolemma is just the name for the plasma membrane of the muscle cell. And in this picture, you see it as this light blue outer covering to the cell. That's just the plasma membrane. We just happen to call it sarcolemma. And it's still doing the usual work. It's holding everything inside, and everything inside is the cytoplasm. But again, the name changes. The cytoplasm is called the sarcoplasm. Okay. So the sarcoplasm is just another name for the cytoplasm, specifically for a muscle cell. What else? It's a normal cell, so we'll find some regular organelles in there. For example, when you look at a muscle cell, you'll actually find lots of mitochondria. Why? Because it takes energy to move, and you remember your mitochondria make energy. You see them as the little orange jelly bean looking structures. They're still the mitochondria. What else? Your muscle cell also has a large endoplasmic reticulum, but we don't break up the endoplasmic reticulum into a rough and smooth in skeletal muscle. And its name is going to change. The endoplasmic reticulum of your skeletal muscle cell is called the sarcoplasmic reticulum. And not only is its name different, its function is different. Remember, we don't break it up into a rough and smooth. Why? Because it's actually not helping to make proteins like rough ER, and it's not really making fats or helping with detoxification like smooth ER. It actually has its own function. I want you to know the function of the sarcoplasmic reticulum. Its one major function is to store and release calcium. The sarcoplasmic reticulum, you can see in this picture, is this large orange lattice work, this whole orange network. See, kind of all around, scattered around. That's the sarcoplasmic reticulum. It's like a big storage container for calcium, because we'll see later on when we talk about how you actually move, calcium plays a huge role in it. So your cells need it around, so they store it in the sarcoplasmic reticulum. But again, other things to see here. Go back to the plasma membrane and look back at the sarcolemma, that blue stuff you see in the picture. If you notice, it looks like some of that plasma membrane, some of that sarcolemma is kind of oozing down into the cell as these little blue tubes. We call those transverse tubules, or a T-tubule for transverse tubule. And that's just an extension of the plasma membrane. And its job is to help conduct the action potential, help conduct some electricity deeper into the cell. And we'll see that a little later on in the chapter. Turns out you're going to need to conduct some of this electricity into the cell to reach the sarcoplasmic reticulum to cause it to release calcium. That's why when you look at the picture, you always see the T-tubule kind of wedged in between neighboring sarcoplasmic reticulums. And we even name this. It forms like a cookie sandwich, an Oreo cookie sandwich, where the cookie wafers or the ends of the sarcoplasmic reticulum, we call them the terminal cisterns. A terminal cistern is just the end to a sarcoplasmic reticulum. And sandwiched in between, like the cream in your Oreo cookie, is the T-tubule. We call this a triad. I think tri for three. A triad is just one T-tubule and two neighboring terminal cisterns. And this structure is going to help to conduct the electricity from the exterior towards the interior of the cell to help reach the sarcoplasmic reticulum. Don't worry, we'll talk about it when we get into the physiology. Right now, just know the parts. A triad is one T-tubule and two terminal cisterns, two ends of two neighboring sarcoplasmic reticulums. What else? There's one more major thing you see inside of the cell. It's what's taking up most of the room. It's these long red rod-like structures in the cartoon. Those are another type of organelle in the muscle cell, and we call it the myofibril. You can see the muscle cell is filled with myofibrils, these long little rod-like structures. And these myofibrils are what we call the contractile organelle, meaning it's the organelle that's going to help you to do your muscle contraction. And by the way, to contract simply means to shorten. So when you contract a muscle, what you're really saying is you're shortening the muscle. And when you shorten the muscle, that will pull on your tendons, which will pull on your bone and help you to move. So when you talk about contractions, even with a female, when she's having uterine contractions during delivery, she's really having muscle contractions. She's shortening muscle in her uterus to help get the baby out. But all this shortening is really the result of this organelle. When the muscle or the cell contracts, when the cell shortens, it's really because the myofibrils have shortened. So we say it's the contractile organelle. It's the organelle responsible for the shortening. That's why muscle cells are crammed filled with these organelles, because muscle cells need to shorten. They need to contract, because that's really how you're going to move. So those are the basic organelles that we see. The sarcolemma is just the plasma membrane forming the outer boundary. Everything inside is called the sarcoplasm, not cytoplasm. It still has nuclei, mitochondria, but then we see other things like the sarcoplasmic reticulum storing and releasing calcium, and myofibrils helping with the contraction. And again, we're going to go in a little bit deeper. This is about muscles, and muscles work by contracting. So we're going to look in on that organelle that helps us to contract. We're going to focus in on the myofibril. And that's what you see here on this picture. You're looking inside of a muscle cell, and the long red rods are the myofibrils. And if you notice, it looks like it has some familiar characteristics. I see some alternating light and dark areas. And we name these areas. It's broken up into repeating segments. And you can see one segment highlighted on the picture. Your myofibril is just made up of repeating units we call sarcomeres. So in the image, they've highlighted one area that represents one sarcomere, and it just repeats over and over, and that entire unit is called the myofibril. So to help explain contractions, we're going to zoom in and focus on a sarcomere and kind of look at what's going on. I want you to know the anatomy, the parts to a sarcomere. Why? Because you can see it's making up the myofibril. So when we say a muscle cell contracts or a muscle contracts, we know it's really because the cell is contracting. And now you know it's really because the myofibril is contracting. We're going to take it one more step. It's really happening because the sarcomere is contracting. So we call the sarcomere the contractile unit, or the smallest contractile unit. It's really the first place you're going to see the shortening or the contraction occur. So we're going to slow down here and kind of spend some time talking about sarcomere anatomy, which is what you see on this image here. Down below, you really see what's making up a sarcomere. It's really a bunch of proteins all held together. That's why muscles really have a lot of protein in it, because the individual parts of it, or at least the major organelle, is made up of lots and lots of proteins. And we're going to focus in just on a couple of the proteins here. For example, some of these proteins help us to highlight the beginning and end to a sarcomere, basically the boundaries of a sarcomere. They're what we call the Z-discs. So on the image, you see on either side is a Z-disc. And it's called a Z-disc because when you look at it, it's this blue line you see, kind of zigzag looking line. That's why it's called a Z-disc. But it really forms the boundaries to your sarcomere. So we say a sarcomere stretches from Z-disc to Z-disc. Those are the boundaries. So you could tell where a sarcomere will start at one Z-disc and it will end at another Z-disc. But that's not really their job. That's just what they happen to do. They happen to be the boundaries. Z-discs are what we call structural proteins. They're proteins that are going to provide structural support for other proteins, meaning they're almost like holding up other proteins. That's what it means to provide structural support. And you can see who it's providing structural support to in the picture. There's these long yellow rods attached to the Z-discs. That's another protein. It has lots of names. It's called actin, and it's a type of thin filament. A filament is just a thread-like protein, and this one is extremely thin, so it's called a thin filament, and its true specific name is called actin. So actin kind of attaches itself to the Z-disc and uses the Z-disc for structural support. What else? Oh, if you notice, there's a protein running right down the middle of the sarcomere. It looks kind of teal in the picture. And it's running right down the middle, almost in a straight line. So we call this protein the M-line. It's another structural protein, just embedded right in the middle of the sarcomere. So we call it the M-line. And again, it's providing structural support, and you could also see who it's providing it for in the picture. You see this long, thick, red rod-like structure attached to the M line. That's yet again another protein. It's really thick compared to the thin actin filament, so it's called a thick filament. And it also has a specific name. It's called myosin. So myosin uses the M line for structural support. And really, how you contract the sarcomere, how you contract the myofibril, the cell, and the muscle is really because of the interaction between myosin and actin. It's really because of the interaction between the thick and thin filaments. So those are the major proteins you're going to see in your sarcomere. Z-discs forming the borders, providing structural support. The M-line in the middle also providing structural support. And then you'll have your thin actin filament and your thick myosin filaments also. And then to finish off pointing out some sarcomere anatomy, we look at areas. We not only name structures, we name areas in anatomy. And so the next things I'm about to name are just areas, regions of the sarcomere. For example, we name the areas where you only have thin filament with no overlap of thick filament. So you can kind of think and look at the picture. Where do you only see actin and no myosin? Only see the yellow bands and no red bands. Turns out those areas are what we call the I band. The I band is just the area where you have only thin filaments with no overlap of thick. And if you look at the picture, you notice the Z discs run right down the middle of your I band. So when we look at a sarcomere, your Z discs have cut the I band in half. So when you look at a sarcomere, you're going to see half of an I band on one side and half of an I band on the other side of your sarcomere. So when we talk about a sarcomere, we say it has two halves of an I band, one half on each side. There's another area to name. It's called the A band. The A band is basically the length of your thick filament. It's the length of myosin. So if you were to draw a line at the tip of myosin on one end and a line on the tip of myosin on the other end, everything in between is called the A band. It just runs the length of myosin. And right in the middle, kind of around the M line, there's an area where you only have thick filament with no overlap of thin. So right in the middle, right around the M line, you see there's only the red myosin filament and no actin filament. We name that area. It's called the H zone. The H zone is just the area where you have only thick filament with no overlap of the thin. So that's basic sarcomere anatomy. Z discs form the borders. It has actin, thin filaments, and myosin, thick filaments. It has the M line right down the middle. It has two halves of an I band, one A band, and one H zone. That's basic sarcomere anatomy. And when you look at the sarcomere, believe it or not, you've seen some of these parts to the sarcomere already. You've actually seen some areas of a sarcomere under the microscope. You've seen the A and I band. How? Well, you see from this table 10.1, under the microscope, when we looked at skeletal muscle tissue, remember one thing we saw were striations. We saw the alternating light and dark bands. Well, when you were looking at striations, you were actually looking at alternating I and A bands. How? Remember, the I bands are where you only have thin filaments. So when you put that under the microscope and turn on the light, it's like you having very thin blinds. So the light's going to come through it really easily, and that's going to look like a light area. And you remember the A band ran the length of myosin, which was thicker. And if you go back to the picture of the sarcomere, the A band will have some overlap of actin in there with the myosin. So it's like you have thicker blinds and extra blinds. So that's going to block a lot of the light. So the A band is going to look darker. So when we were looking at skeletal muscle tissue under the microscope in lab before the break, you were actually looking at alternating A and I bands already. You just didn't know it. And so when you look at a muscle, again, I told you muscles have lots of proteins, and it's all because of all the proteins you see within your sarcomere. And again, we've seen these two already. We've seen two of the proteins that are responsible for the contractions. That's why we call them contractile proteins or contractile filaments. They're actin and myosin. So when we talk about the physiology of how you move, we're going to really be talking about how do actin and myosin interact. And remember, this is skeletal muscle tissue. So they're only going to interact when you allow them to. So we need something to prevent them from interacting unnecessarily. We need something to help us to regulate when actin and myosin can interact. Turns out that's the job of other proteins in your muscle cells. We call them regulatory proteins. There's going to be two major ones. They're called troponin and tropomyosin. They're called regulatory proteins because they're going to help to regulate when myosin and actin can interact. How? Well, if you're not moving, well, you don't want actin and myosin to interact. And the way you're going to do that is that troponin and tropomyosin are actually going to cover actin. You can see from the picture down below, troponin in the cartoon is the little blue looking structures, and tropomyosin is the long golden rods. And to help regulate contractions, they actually sit on top of actin and block myosin binding sites, which are just sites on actin where myosin will need to bind when they interact. So to keep you from moving, troponin and tropomyosin block the myosin binding sites on actin. And when you want to move, those two proteins move away from the myosin binding sites and allow them to interact. So we'll talk about how that's going to occur again when we get to the physiology. Right now, we're just letting you know that these things exist. And like usual, these are not the only proteins. There are lots of other proteins, especially structural proteins, helping to hold all these things together. You do not need to know these structural proteins in the box. You do not need to know titin, nebulin, alpha-actin, myomycin, or dystrophin. You do not need to know those structural proteins. But that's pretty much it on the microscopic anatomy of important structures in the muscle cell. Remember, like usual, trust the exam review. And so the rest of this chapter is switching in finally into the physiology. We're going to talk about how do you move. And like usual, there's going to be lots of steps in this, just like how there were lots of steps in the generation of an action potential. There are going to be lots of steps involved with how do you generate a muscle contraction? How do you move? And I want you to know the steps in order for the generation of a muscle contraction. How do you move? Excuse me. And we're going to go through them. And just to help you to save some time, a lot of these steps are highlighted already in the slide on your PowerPoint. But I'm just going to go in order of the slides that are here. And unfortunately, in this order, which is the order it's given in your textbook, they actually don't start in the beginning. They actually start towards the end. And they start off basically when you're in the process of moving. Remember, when you're moving, you're really just shortening the muscle. And you're shortening the muscle by shortening the cells. And you remember the cells are shortening because the myofibril is shortening. And you remember the myofibril is shortening really because sarcomeres are shortening. And you know now sarcomeres are shortening because actin and myosin are interacting. So we're starting here. We're starting where actin and myosin interact. And we help to explain how actin and myosin will interact in what we call the cross-bridge cycle, or the sliding filament model or mechanism. It's called the sliding filament model because they're basically looking like they're sliding across each other, kind of like how your fingers look when you interlock them. If you were to slide them to interlock your fingers, that's a lot like how the sliding filament model of how actin and myosin acts will represent. How so? Well, it's really because myosin is pulling on actin, like a game of tug of war. Myosin is going to actually pull actin towards the M line, towards the middle. And as it's pulling on actin, actin will pull whatever it's attached to. And remember, actin's pulled or attached to the Z disc. So when actin's being pulled by myosin, one thing we're going to see when we look at it is that the Z discs will actually look like they're being pulled towards each other on either side of the sarcomere. It's really because they're being dragged because they're attached to the actin, which is being dragged towards the middle of the cell. But again, keep it simple. We're going to go through the steps. It's really four major steps to help to explain the sliding filament model. And again, not only is actin and myosin going to move, but what's attached. And I told you, we're going to see this in the sarcomere. And you can see that on this picture here. This picture is just showing us all the changes we're going to see within a sarcomere as the muscle, the sarcomere, the myofibril contracts. And again, just think about what everything's attached to. I told you myosin's going to pull on actin, and actin's attached to the Z disc. So one thing that happens in a sarcomere as you contract is that the Z discs will move towards each other. They will move closer together. That's one thing that happens in your sarcomere during a contraction. Z discs move closer together. What else? Well, as myosin is pulling on actin, they're going to begin to overlap. Remember, they're sliding across each other. Why is that important? Well, you remember there are areas in your sarcomere where there is no overlap, where there's only thin filaments or only thick filaments. Well, we're overlapping now, so you're going to begin to lose some of those areas. Remember, one area was called the eye band. Remember, the eye band is where you only have thin filaments. On this cartoon picture, it's represented by the light blue box. That light blue box is representing the area we call the eye band. And as you contract, well, myosin and actin are going to slide and overlap, so you're going to lose where you only have thin filaments. So during your contractions in a sarcomere, we say the eye band will narrow and possibly disappear. Why? Because myosin and actin are overlapping. Remember, there was also an area where we only had the thick filament, myosin. Remember, that was called the H zone. Well, again, as you're contracting, myosin and actin are overlapping. They're sliding against each other. So you're going to also lose that area of where you only have thick filaments. So just like the eye band, the H zone, when you contract, will narrow and also possibly disappear. Those are the three major things I want you to know that happen in a sarcomere during a contraction. One more time. Z discs move closer together. Eye bands will narrow and possibly disappear, and H zones will narrow and possibly disappear. That's what happens. What happens to the A band? Remember, the A band runs the length of myosin. And did I mention anything about myosin getting shorter? No. Well, the A band is going to stay the same. That's why I didn't mention the A band. Okay. So that's what happens in your sarcomere during a contraction. And how? How do actin and myosin interact? What's the cross-bridge cycle? What's the sliding filament model? Well, it's really just 4 steps, 4 simple steps. And these 4 steps are basically the last 4 steps in the generation of a muscle contraction. It's how you shorten. And you remember, first thing that actually has to occur is that you need to remove those regulatory proteins. So let's assume that myosin and troponin have already been moved. And how does that happen? It's super simple. It's calcium. To get troponin and tropomyosin to move, all you need is calcium to bind to troponin. And when calcium binds to troponin, troponin and tropomyosin will move away from the myosin binding sites on actin. Kind of think of troponin as the boss. So you only have to talk to troponin and it will tell tropomyosin what to do. So calcium will bind to troponin, causing troponin and tropomyosin to move away from myosin binding sites on actin. So we're going to assume those steps have already occurred. So these 4 steps I'm about to mention are this cycle, this cross-bridge cycle, this sliding filament model. First step is something that myosin needs to do. If you notice from the picture, you see the yellow beads up top. Those are representing actin. And this long red rod-like structure with these bulbous heads coming off is myosin. And it looks like actin is kind of hovering over myosin. And those little bulbous structures, what we call myosin heads, they need to look up at actin. They need to cock their heads back. And in order to do that, they need to use ATP. When you use ATP, we say you hydrolyze ATP. So the first step is that myosin heads hydrolyze ATP to form the cocked position. AKA, myosin heads will use ATP to cock their heads back to look up at actin. Then once they do that, it's time to move on to step 2, which is for myosin to bind to actin. They need to touch. Remember, they're going to pull on actin in a game of tug of war. And in order for you to pull, well, you need to attach. So step 2 is that the myosin heads will bind to actin. And when the myosin heads bind to actin, they form a bond, which we call a cross bridge. So that's step 2. Myosin heads bind to actin, forming a cross bridge. And then once they bind, now they could pull. And when myosin heads pull on actin, we call that the power stroke. And you remember, they're going to pull them towards the center of the sarcomere. They're going to pull them towards the M line. That is step 3. In step 3, myosin heads pull actin towards the M line during the power stroke. But again, this is like a game of tug of war. You can't pull the rope once and win the game. You gotta pull, then let go of the rope with one hand, then reattach to it, then pull again, then let go with one hand, then reattach and pull again. Your myosin heads need to do the same thing. Once they pull once on actin, they need to let go so they could grab and pull again. So the last step, the fourth step, is how the myosin heads are gonna let go of actin. And it's super simple. To release actin, myosin heads simply bind ATP. That's it. They don't even use it. They just bind to it. And when myosin heads bind to ATP, that will cause them to release actin. And then this cycle will resume again. That ATP the myosin heads bound to release actin is actually the ATP they're going to use or hydrolyze to form the cocked position. And this cycle will occur multiple times until you've reached maximum contraction, until you've flexed or contracted the muscle, its maximum possibility. So those are the steps. They happen over and over until you hit maximum muscle contraction. One more time. Myosin heads hydrolyze ATP to form the cocked position. Then they bind actin, forming the cross bridge. Then they pull actin towards the M line in the power stroke. And then they bind ATP to release actin. Then repeat. Myosin heads hydrolyze that ATP to form the cocked position, bind actin to form the cross bridge, pull actin towards the M line in the power stroke, bind another ATP to release it. Then they will use the ATP that form the cocked position, bind to actin to form the cross bridge, pull actin towards the M line to form the power stroke, and then bind another ATP to release it, and repeat over and over again until you've hit maximum contraction. So this is basically how your cells are contracting, how the myofibrils in the cells are contracting, the cells themselves are contracting, the muscle is contracting, and that's really how you're moving. These are the last 4 basic steps to a contraction. But again, there's more to know. I told you those were the last 4 steps. So we got to go way back to the beginning. And it actually begins with nervous tissue. You know, if you want to walk to the door, it begins with you deciding to walk to the door. And once you decide, it's going to begin with your brain sending signals via neurons to the neurons in your spinal cord, which will send that information to neurons and nerves in your body, motor neurons, which will then talk to the muscle cells. And remember, one way your neurons are communicating is via electricity. So it's really some sparks of electricity that will lead to an eventual contraction. It's really these excitable cells. Remember, neurons are excitable cells. They can make an action potential. Turns out, once a muscle cell gets a signal from a neuron to contract, it will also make a spark of electricity. Remember, this is why we did nervous tissue first. Remember, as soon as your neuron releases its neurotransmitter, you could get either an excitatory postsynaptic potential in the recipient cell or an inhibitory postsynaptic potential in the recipient cell. Well, we're talking about muscles. We're talking about stimulating them for movement. So your muscle cell, once it gets a signal from a neuron, will generate an excitatory postsynaptic potential, which will cause it to depolarize. And if it gets a strong enough signal, it will hit threshold and make its own action potential. Pretty similar to how a neuron does. So not only are neurons excitable cells, so are muscle cells. And we're going to see as we go through these steps in the generation of a muscle contraction, your muscle cells at one point will make electricity. So we name this combination between your cells making electricity, which will eventually lead to your actin and myosin sliding across each other. We call this the excitation-contraction coupling theory. It's how we're going to talk about electricity and relate that somehow to this contraction, or this coupling of a contraction in your muscle cells. And when we talk about muscle cells and this coupling, like usual, there's going to be an optimum for everything. As in biology, there's optimums for when and how you can contract. And it has to do with the sarcomere, We saw in the sarcomere, there were lots of these random areas. Things were spaced out in a sarcomere when you're relaxed. And as you're contracting, things get shorter and squeezed tightly. There's a reason for this separation initially. It's because there's a relationship between the length of your muscles and cells and how much force you could generate. Turns out, this is what we call the length-tension relationship. The amount of force your muscle cells will generate will depend on the length of the sarcomeres. So if your sarcomere is already contracted a little bit, well, you're not going to have that much room to do your sliding filament model. You're not going to generate that much force. But on the other hand, if your sarcomere is stretched out too far, well, it might be too hard for actin and myosin to interact if they can't even reach each other. So you might not even get a contraction if the muscle is stretched too far. So there's this slide in the middle area where you're going to have an optimal contraction. Why? Because things are nice and evenly spaced out, giving them lots of room to do this sliding filament model. No, you do not need to know this for your exam. Like usual, stick to the exam review. We want to focus more, though, on the steps of a muscle contraction. And now we're going to finally start at the beginning. And we're going to bring it down to the level of the muscle cell. How do you stimulate it? Well, we know from our previous chapter that we did, chapter 12, that's going to begin with a neuron. Remember, there are neurons specifically designed to tell muscles what to do. Remember, we call them motor neurons. And remember, neurons typically communicate with other cells using their axon terminal. So when we talk about how a neuron stimulates a muscle cell, we're really talking about how a neuron will use its axon terminal to communicate with a muscle cell. And you know some things already. You know the name of the place where a neuron will communicate with another cell. Remember, we call that a synapse. Remember, a synapse is just a place where a neuron will communicate with another cell. And we're still talking about a synapse. But like usual, this name is going to change. We're talking about a specific synapse. We're talking about the synapse where a neuron will talk specifically to a skeletal muscle cell. We call this a neuromuscular junction. It tells you in the name, neuro for neuron, muscular for muscle. Remember, junctions are cellular junctions. They're between cells. So a neuromuscular junction is just a synapse where a neuron will talk to a muscle cell, where a neuron will stimulate a muscle cell. So like usual, we got to go through some anatomy. And that's what you see on this image here. It's showing some anatomy of a neuromuscular junction. Remember, it's just a synapse. You know the three basic parts of a synapse. Remember, one part is the axon terminal of your neuron sending the signal. That's what you see in the picture off to the right. They've zoomed in, and that yellow structure up top, that's the axon terminal of your neuron. Remember, another part to your synapse was the plasma membrane of the cell receiving the signal. In this case, it's the plasma membrane of the muscle cell, skeletal muscle cell. Remember, that's the sarcolemma. But yet again, the name is going to change. When we talk about the plasma membrane of a skeletal muscle cell, specifically at a neuromuscular junction, we call it a motor end plate. A motor end plate is just the plasma membrane of your skeletal muscle cell, specifically when you're talking about a neuromuscular junction. And remember, when your neurons talk to a cell or communicate with another cell, they don't actually have to touch. Remember, there's a space in between. Remember, we call that space the synaptic cleft or the synaptic space. It's still there because this is still a synapse. So these pictures are just showing you a neuromuscular junction is a synapse just like any other. It'll have those three parts. It will have an axon terminal, where you'll have your chemical messenger inside, your neurotransmitter, and you'll release it to help bind receptors on the muscle cell. And so let's begin charting this journey in a muscle contraction. If you wanted to contract, it will begin with an action potential arriving at the axon terminal of your neuron, of your motor neuron. Why? Why do you need that spark of electricity to arrive? Well, that spark of electricity, that action potential, when it arrives at the axon terminal of your neuron, it's actually going to open some voltage-gated channels. When this spark of electricity, this action potential arrives at the axon terminal, it's going to change the charge across the membrane. And when you do, remember, that will affect some voltage-gated channels. In this case, it's going to cause calcium voltage-gated channels to open. And when these voltage-gated calcium channels open, calcium is going to rush into the cell. And when calcium rushes into the cell, that will trigger the release of acetylcholine, which is our neurotransmitter, which is our chemical messenger. So that's the first step. First step is the action potential arrives at the axon terminal, triggering the opening of voltage-gated calcium channels, which will allow calcium to rush into the axon terminal and trigger the release of acetylcholine. Then once you release the acetylcholine, it has to make that journey across that gap in the middle. It has to make the journey across the synaptic cleft. And it's going to do that using diffusion. It's going to go from where there's a lot of them, which is closer to the axon terminal side, to where there's fewer of them, which is closer to the motor end plate side. So in step 2, we just simply say acetylcholine diffuses across the synaptic cleft. It drifts across the synaptic cleft, and then it will bind to receptors on the motor end plate. That's step 2. Step 2, one more time. Acetylcholine diffuses across the synaptic cleft and binds receptors on the motor end plate. And you're still not moving yet. There's still more steps to go. You already know the last steps. We're not even there yet. As soon as the muscle cell gets its signal, gets the acetylcholine to bind its receptors, remember, this is an excitatory neurotransmitter. We know that already. We're going to generate an excitatory postsynaptic potential in our muscle cell, which is going to cause it to depolarize, aka become slightly positive, until it hits threshold, and it will generate its own action potential. And when it generates its own action potential, that action potential will move as well. It will travel along the entire plasma membrane of the muscle cell. Why is that important? Because you remember, parts of the skeletal muscle's plasma membrane dives deep into the cell. Remember, we call that a T-tubule or transverse tubule. So when the action potential travels along the entire plasma membrane, it will even travel down a transverse tubule. Why? This is finally why that triad comes into play. Remember, a triad has a T-tubule, and on either side of it are sarcoplasmic reticulums. There's a sarcoplasmic reticulum sitting right next to the T-tubule. So when that action potential travels down the T-tubule, it's kind of going to zap the sarcoplasmic reticulum. And the sarcoplasmic reticulum is like this one friend. Everybody has this one friend. Who's this one friend? Everybody has this one friend where whenever you scare your one friend, they fart. Just a little bit, just a little bit. Everyone has that one friend, when they get scared, they fart just a little bit. If you don't, maybe it's you. I don't know. But anyway, the sarcoplasmic reticulum is kind of like that one friend. When the action potential zaps it, it's almost like it farts. But in this case, it's farting out calcium. So the action potential, when it travels down the T-tubule, will trigger the release of calcium from the sarcoplasmic reticulum. And once the calcium gets released from the sarcoplasmic reticulum, it's going to travel throughout the entire cytoplasm, looking for one thing. You know what one thing from our sliding filament model. It's going to look for troponin, and it's going to bind to troponin, causing troponin and tropomyosin to move away from those myosin binding sites on actin. And you know the rest. This one slide shows us all the steps to our skeletal muscle contraction. Let's go through them all one more time, slowly. So the first step is that your action potential will arrive at the axon terminal, which will trigger the opening of voltage-gated calcium channels, and calcium will rush into the axon terminal, triggering the release of acetylcholine. That's step 1. Then step 2 is acetylcholine will then diffuse across the synaptic cleft and bind receptors on the motor end plate. And then that will trigger step 3, which is when your muscle cell will depolarize and generate its own action potential, which will travel along the entire plasma membrane, aka sarcolemma, and down a T tubule to trigger the release of calcium from your sarcoplasmic reticulum. Then step 4, calcium will bind to troponin, causing troponin and tropomyosin to move away from the myosin binding sites on actin. Then step 5, myosin heads will hydrolyze ATP to form the cocked position. Step 6, myosin heads will bind to actin, forming the cross bridge. Step 7, myosin heads will pull actin towards the M line during the power stroke. And step 8, myosin heads will bind ATP to release actin and cycle again and again and again until you've hit maximum contraction. That's how you move. This one slide does it all. Action potential arises at the axon terminal, triggering the opening of voltage-gated calcium channels where calcium will rush into the axon terminal, triggering the release of acetylcholine. Acetylcholine will drift or diffuse across the synaptic cleft and bind receptors on the motor end plate, which will cause the muscle cell to depolarize and generate its own action potential, which will travel across the entire plasma membrane and down a T tubule, where it will trigger the release of calcium from the sarcoplasmic reticulum. And then that calcium will then bind to troponin, causing troponin and tropomyosin to move away from the myosin binding sites on actin, while myosin heads will hydrolyze ATP to form the cocked position. It will bind to actin, forming the cross bridge. Myosin heads will pull actin towards the M line during the power stroke. Then myosin heads will bind ATP to release actin. And that's how you're going to move. Those are all the steps in the generation of a muscle contraction. Luckily, you don't have to think about any of that besides just wanting to move. But that's how you move. But then you got to consider, how do you relax? Turns out you got to do 2 things to relax. One of them is really attacking the first steps. It's attacking acetylcholine. For you to relax, you got to stop stimulating the muscle cell. And you're going to stop that by getting rid of a neurotransmitter. You even know how to do that. Remember, there are 3 ways to get rid of a neurotransmitter. Which of those ways are you going to use to relax your skeletal muscle? Turns out you're going to use enzymatic degradation. You're going to break it down with an enzyme. And we even know the enzyme that we use to break down acetylcholine. It's in the picture. It's called acetylcholine esterase. Acetylcholine esterase will literally break down and destroy acetylcholine. And so if there's no acetylcholine, your muscle cell will not get stimulated and you'll be able to begin relaxing. But to finish relaxing, you got to do one more thing. You got to get rid of calcium. Remember, calcium is allowing your regulatory proteins to move out the way. You have to put them back. So in order for troponin and tropomyosin to go back to binding to actin, you got to get rid of the calcium. How? You put it back into its sarcoplasmic reticulum. And to pump it back, you're using pumps in the membranes of the sarcoplasmic reticulum that require ATP. So it turns out not only do you need ATP to move, because you need ATP for myosin head to form the cocked position and to release actin, you even need ATP to relax. You need ATP to put the calcium back into the sarcoplasmic reticulum. This is why muscles use so much energy. They not only need energy to move, they even need energy to relax. And because of that, we're going to have to spend some time talking about that. We're going to have to spend some time talking about the sources of energy for your skeletal muscles. Turns out there are 3 major sources of energy for your skeletal muscles and your skeletal muscle cells. What are they? They're listed on the slide. There's something called creatine phosphate, anaerobic glycolysis, and cellular respiration. Those are your 3 major sources of energy for your skeletal muscles. Let's go through them one by one. Starting off with the one that gives you the energy for the least amount of time. Turns out you have 3 sources of energy because they all give you energy for differing amounts of time. And the first one I'm mentioning is only going to give you energy for a couple seconds. It's called creatine phosphate. So how's creatine phosphate giving you energy only for a couple seconds? Well, it has to deal with ATP's structure. Oh, we got to dig way back to our structure of ATP, way back to chapter 2. Remember, ATP has 3 phosphate groups in it. Remember, it's called triphosphate because it has 3 phosphate groups. And remember, the energy is in the bonds really between the first and second phosphate group and the second and third phosphate group. So let's say you ate a Whataburger cheeseburger, and odds are if you're at home right now, you probably ate more energy's worth of food than you're using right now. And so your body's going to try to save some of that energy. What your body's going to do is it's going to take ATP and it's going to take creatine. And it's going to take a phosphate. It's going to pop off that third phosphate group and give it to the creatine to form the creatine phosphate. And then let's say you go out for a walk. Now you're going to need energy to power your walk. Now you're going to take that phosphate group from creatine phosphate and attach it to a lesser form of ATP called ADP, which is adenosine diphosphate. Di for 2. It only has 2 phosphate groups on it. And you're going to take that phosphate from creatine and attach it to the ADP to form ATP, which is how you could use the energy. So creatine phosphate is basically a storage container for energy because it's storing a phosphate group. That's how it's giving you energy. And you could use it up. You could use up all your creatine phosphate. And once you use it up, you're no longer getting energy from it. And you use it up quite quickly. It only gives you energy for a couple seconds. So when you jump on a treadmill, the first couple seconds of you on a treadmill feels super easy because you're just using stored energy. You're just using up your creatine phosphate. And that's only for a couple seconds. But we know we could usually do a little bit more exercise than a couple seconds, or at least I hope we can. So for us to power the rest of our workout, well, we're going to need another source of energy. Another source of energy is thanks to anaerobic respiration, sometimes called glycolysis or sometimes called anaerobic glycolysis. All synonyms. Anaerobic glycolysis or just glycolysis or anaerobic respiration. This is another source of energy. But this one is now only going to give you enough energy for a couple minutes. How? Let's go through it. It's called anaerobic because it does not require energy. This is a process of, does not require oxygen. This is a process that makes energy without oxygen. How? The next part of the name gives you a hint. It's called glycolysis. Glycolysis is the process of breaking down sugars for energy. So when you say anaerobic glycolysis, you're basically saying I'm breaking down sugar for energy without oxygen. And that's all this is. That's why when you get a little hangry, what do you reach for? You reach for a Snickers so you can break down all the sugars in the Snickers for energy. That's basically what this is. You're breaking down glucose for energy without oxygen. And because you're not using oxygen, this is not going to be a very efficient way to make energy. So you're not going to make that much. You're only going to make enough for maybe a couple minutes. Kind of imagine trying to work out while holding your breath. It's practically impossible because this is not giving you enough energy. But no worries. There's yet again another source of energy. This last source of energy is going to give you the most energy. So it's going to give you energy for hours. It's going to give you the longest amount of energy. It's called cellular respiration, or sometimes called aerobic respiration. It's still a type of respiration where you're breaking down sources of food for energy. In this case, yes, it could be sugars, but not just that. Think major food sources. There are carbs, sugars. There are proteins and amino acids. And there's fats and fatty acids. Remember, we talked about these major organic compounds. You could break them all down for energy. But in order to do that efficiently, you're going to need some oxygen. And because you need oxygen, we call it aerobic. So aerobic respiration is when you break down organic sources of fuel for energy using oxygen. And this is basically what your mitochondria do. This is why your mitochondria are the powerhouses for your organelles. This is why they're the nuclear power plants. This is why they make so much energy, because they're doing aerobic respiration or cellular respiration. They're breaking down food sources for energy using oxygen. And this helps to explain your life. This is why whenever you exercise, whenever you hop on that treadmill, first couple seconds, it feels a breeze as you're just burning through all your creatine phosphate. Over the next minute or so, you're still probably doing okay, but you're probably feeling a little sweat start to form. But then afterwards, you notice you're huffing and puffing. You're breathing hard and fast when you're working out. Why? Whole point of you huffing and puffing when you're working out is to help provide oxygen to your mitochondria so that they could do aerobic respiration. Why? Because this is going to be the source of energy that's going to give you the most energy to help power your body for the longest amount of time. This is why you breathe harder and faster as you work out longer. You're just trying to keep up with the oxygen demand of aerobic respiration occurring in your mitochondria. So those are your three major sources of energy. But if you've ever worked out, no matter how much energy you might pack into your body, eventually you're going to hit the end to your workout. No matter how much food you eat, you eventually will get tired and not be able to work out. You're going to hit what we call muscle fatigue. Muscle fatigue is literally your muscles' inability to contract. They cannot contract. Why? Because they don't have enough energy. Just think about it. Maybe you ran out of your creatine phosphate, or there's a point in your workout where no matter how fast you're breathing, you're not going to take in enough oxygen. No matter how hard or fast you breathe at one point in your workout, you're not delivering enough oxygen to your cells. And they're not going to be able to generate enough ATP, and you're not going to be able to contract. So kind of think of running out of energy will cause a muscle fatigue. You've probably seen this when you've watched marathons or commercials of marathon runners. Sometimes you see a marathon runner make it to the finish line, and right before they get to the finish line, they collapse. And it's like they try to crawl. You know the person wants to make it to the finish line, but their muscles just won't let them. You're literally watching someone hit a muscle fatigue. They have literally run out of energy. Almost like your car running out of gas. You could run out of gas too. In this case, energy. Why? Because I told you there's going to be a point in your workout where no matter how hard you breathe, you cannot take in enough oxygen that your body requires. You're going to hit what we call an oxygen debt. An oxygen debt is a debt. When you owe something, you owe more oxygen than you really are getting. And when you owe this oxygen, well, your body's not going to make ATP. It's waiting for the oxygen. You're not going to move. So this is one major reason why you cannot work out indefinitely. And knowing this stuff, remember, we could take advantage of it. This helps to explain stuff. Like sporting events. If you've ever watched, for example, a football game, you might notice sometimes on the sidelines, the players have on gas masks. It looks like they have a little oxygen tank with them. It's because they do. It's because they're trying to outpace this oxygen debt. They're just literally inhaling this pure oxygen from this canister to help keep up with the oxygen demands of their cells as they're playing the game. So when we know what happens in anatomy, we could take advantage of it to our benefit, like usual. So that helps to explain the basic contractions of a muscle and how do you get the energy to help power it. So the rest of this is kind of switching gears and talking about more of the forces that your muscles can generate. Now that we know how to contract, now we could talk more about the forces of the contraction. For example, we could talk about how do you control muscle tension. And when we talk about muscle tension, think of it as the strength of a contraction. Meaning I can make a big contraction, generate a large strength. Kind of imagine trying to pick up a 300-pound dumbbell. You're going to have to generate a lot of strength, or you're going to generate a lot of tension to try to pick it up. Versus trying to pick up a feather. Feathers are practically weightless. So to pick it up, you have to generate very little tension, very little force, use very little strength. So we're going to talk about how do you do that? How do you control the tension that you generate? How do you control the force of your muscle contraction? Well, think about the first step in the generation of a muscle contraction period. It starts off with neurons. It starts off with the nervous system. Turns out one neuron could stimulate more than one muscle cell. And we give the whole thing a name. And remember, we even named the neuron talking to muscle cells. Remember, the neuron responsible for stimulating muscle cells are motor neurons. So we actually give a name to a motor neuron and all the muscle cells, aka the muscle fibers, it will innervate, meaning it will stimulate. We call this a motor unit. A motor unit is one motor neuron and all the muscle cells it stimulates. And to help control your tension, you just simply control the number of motor units. For example, if I want to pick up a feather, I don't need my entire muscle strength to do it. I only need to stimulate maybe a handful of cells to pick up a feather. So I'm really going to use maybe 1 or 2 motor units. I'll probably use 1 or 2 neurons to talk to a couple of the cells to help me pick up that feather. But if I have to pick up a 300-pound dumbbell, well, I need my entire muscle to work now. And for my entire muscle to work, I need to stimulate all the cells. So I need more neurons to talk to more of the cells. I'm going to need more motor units. So this is how you're controlling your contraction strength, your muscle tension. It's just you using fewer motor neurons, fewer neurons in the cells in your muscle, or more of them. That's all that is. And if you're wondering, how is it that we know all this stuff about muscle cells? It's because we study them. And it all actually begins very simply. We learn a lot of information just by studying one cell and studying one cell's single contraction. A single muscle contraction of a single cell is what we call a twitch. It's a brief contraction of muscle cells. And when you look at a twitch, a brief contraction, you still see all the steps. It has to go through all those steps we talked about in the generation of a muscle contraction. And when you look at all the steps in the generation of a muscle contraction, it can all actually get broken down into 3 major periods, which is what you see on this chart. They're all highlighted in different colors. There's something called the latent period in blue, the contraction period in red, and the relaxation period in green. Those are the 3 major periods in a contraction. They're related to the steps. The latent period is when the muscle cell has not yet gotten the signal, where there's movement that's about to occur, but the muscle cell doesn't know it yet. How? Well, think about your first steps. This is like when the action potential is arriving at the axon terminal. The muscle cell doesn't know that. That's happening in the neuron. When you're opening voltage-gated calcium channels and allowing calcium to rush in, the muscle cell doesn't know that. That's happening in the neuron. When acetylcholine is getting released from the axon terminal, the muscle cell doesn't know that yet. This is all happening in the latent period. So you're not going to see a contraction because the muscle cell doesn't know yet. It's waiting to receive the signal. But once it gets the signal, well, it's going to go through the steps of a contraction. That's what you see in red. And then once it's done contracting, well, it needs to relax. That's in green. Oh, and if you notice, it spends a large time relaxing. Almost half the time you see in the chart is spent relaxing. Why? Because it takes time. You got to think about the things you have to do to relax. It takes time to destroy all of the acetylcholine that was released from the axon terminal. It's going to take time to gather up all that calcium and put it back into the sarcoplasmic reticulum. So believe it or not, your muscle cells spend a large time relaxing, kind of going back and resetting to get ready for another muscle contraction. Remember, like always, stick to the exam review. What else? We talked about how do you control the force. Not only can you control the force, you could control how long you hold the contraction. Meaning I could hold a contraction. I could pick up a weight and hold it for a period of time and then decide to put it down. When you hold a contraction, a sustained contraction is when you hold a contraction We call that a tetanus. A tetanus in anatomy is when you have a sustained muscle contraction. So how do you do that? How do you generate a tetanus? How do you hold a contraction? Well, again, it has to deal with the neuron. You see from this chart here, it's called a myogram. It's just showing in red the force of your contraction. The higher up the line goes means the stronger the force. And the flat plateau you see of the red line way over to the right is someone holding a contraction. It's a tetanus. How do you do that? How do you hold a contraction? Well, it has to deal with the second line on the chart in blue down below. Blue is representing all the action potentials that are being fired off from your neurons. And you notice as you read it from left to right, a single action potential could cause a slight twitch, a single generation of a force. And then that muscle cell will go back to relax. But if you notice, if you were to fire off an action potential, and then immediately fire another one, the muscle cell does not have time to relax. That's what you're seeing in the B area. Before that muscle cell could finish relaxing, another action potential has stimulated it to contract again. And that second contraction, because it has not quite relaxed, is a bigger one. So every time the neuron fires off an action potential, the muscle cell will contract. And if it's not given time to relax, it will contract harder. And if you get enough of these action potentials, it could hold the contraction. So if you were to put your hand into a ball and kind of close your fist up and squeeze and hold it, you're holding that contraction because your neurons are constantly firing off at the muscle cell to keep it contracted. And when you relax, it's because you stopped firing the action potential and you've gotten rid of acetylcholine and you've put your calcium back into the sarcoplasmic reticulum. And again, not only can you hold a contraction, remember, we could change the force. This is just like us again talking about the motor unit. Remember, a motor unit is just a motor neuron and all the muscle cells it will stimulate. And remember, if you want a very weak contraction, you just want to move a little bit or generate a weak, brief force, you're just going to use very few motor units. But if I want to have a long, sustained contraction or generate a great force, I'm going to increase the number of motor units. And we even give a name to that. When you increase the number of motor units, either to sustain a contraction or increase the force of a contraction, we call that motor unit recruitment. Motor unit recruitment is just when you increase the number of motor units to either sustain a contraction or increase the force of a contraction. And like always, luckily, we're not thinking about this. This is things our muscle cells are doing on our behalf. What else? Turns out you know a lot about skeletal muscle cells and skeletal muscle tissues now. So now we could talk about some exceptions to some rules. Turns out I kind of lied to you at the beginning of this chapter. How? Well, I told you we're talking about skeletal muscle. And I told you at the beginning of this chapter, you do control skeletal muscle. It's voluntary. Well, it turns out not all the time. Sometimes you have involuntary contractions of skeletal muscle, meaning sometimes skeletal muscle will move without your permission, and it happens quite often, and we give a name to it. It's called muscle tone. Muscle tone is just when you have involuntary contractions of skeletal muscle, but for good reasons. Why? When do you have muscle tone? Well, it turns out you have a lot of muscle tone in your back muscles. The muscles in your back are skeletal muscles, so you could use them if you wanted to, but it's also contracting without you thinking about it. How do you know? Well, it turns out if the muscles in your back were to relax, you would fall and slump over due to gravity. But because your muscles are contracting, you're either sitting up straight or you're standing up pretty straight. Why? Because they're contracting without you thinking about it. You're having muscle tone in the skeletal muscles in your back. So yeah, if I ask, skeletal muscle is still voluntary, but there is something called muscle tone. There are occasions when it can contract involuntarily. What else? Oh, we know how a muscle contracts, but you might not know that there are also different types of contractions. Turns out if you wanted to work your muscles out, you have multiple ways to do it because there are multiple types of contractions. And I want you to know these two major types of contractions. There's an isotonic contraction and an isometric contraction. Let's go one by one. Like usual, don't be intimidated. The names give you a hint. Let's start off with isotonic. Iso, just break the name down. Iso means the same. Tonic is for tone, as in tension, as in the force your muscle can generate. So in an isotonic contraction, the force or the tension will stay the same, but the muscle length will change. That's an isotonic contraction. It's when the force or the generation stays the same, but the muscle length changes. This is actually what you think of when you think about lifting weights. Lifting regular weights, think of doing bicep curls. Imagine having a 20-pound dumbbell. And when you're doing your bicep curls, that 20-pound dumbbell in your hand doesn't change. Doesn't matter if you bring the dumbbell up closer to the shoulder or you bring it down closer to the hip and knee. When you're doing your bicep curls, you constantly have to hold this 20-pound dumbbell. So you will constantly generate this force to help you to hold this 20-pound dumbbell. But as you're doing your bicep curl, when you bring the dumbbell up, where you're contracting the muscle, you're shortening the muscle. And when you bring the dumbbell down, you're lengthening the muscle. So the muscle length is changing, but you're still generating the same force to hold your dumbbell. You're doing isotonic contractions when you lift weights. And this is anatomy. We name everything. We even give names to the different time periods when the muscle length changes. We give a name to when the muscle length is long and when it's short. When you shorten the muscle length during your isotonic contraction, we call that the concentric portion. The concentric portion is when you shorten the muscle. And then the eccentric portion of your isotonic contraction is when you lengthen the muscle, when it gets longer. So that's your isotonic contractions. And then there's one more way. If you don't like lifting weights, don't worry. There's another way to work out because there's another type of contraction. It's called an isometric contraction. Again, don't be intimidated. Just break down the name. Iso still means the same. Metric as in a unit of measurement, possibly for length. So think length for metric. So in an isometric contraction, now the muscle length will stay the same, but the force or the tension will change. So isotonic and isometric are basically opposites. In an isometric, the muscle length will stay the same and the force or the tension will change. What's an example of an isometric contraction? Well, an example is what you see on the next slide of this young man holding a book. All the way to the right in segment C, that's an isometric. He's holding that book in his extended hand. And I could force him to generate a bigger force if I just added another book on top without him moving his arm. That's a type of isometric contraction. Or another example of an isometric contraction is pushing against a wall. You could push against a wall hard with as much force as you want, or you could push against the wall gently. But you don't necessarily have to move your arm as you're pushing against the wall. So your muscle length is not going to change, but the force you use can change. That's an isometric contraction. Again, keep it simple. Isotonic contractions, the tension or the force stays the same while the muscle length changes. And when the muscle gets shorter, call that concentric, and longer is eccentric. And then isometric contraction is the opposite. It's when the muscle length stays the same and the force or the tension will change. That's it. That's your different types of contractions. And again, there's a couple other things to talk about. Not only do we know the different types of contractions, we could actually go back microscopically again and look at the skeletal muscle cells. You know what a skeletal muscle cell looks like already. So now we could complicate it. Turns out, not only do the different types of muscle tissues differ, but cells can differ. When we look at the different skeletal muscle cells, they are not all the same. Turns out there are 3 major types of skeletal muscle cells. Remember, a muscle fiber is just another name for a muscle cell. And there are 3 major types. There are slow oxidative muscle fibers or muscle cells. There are fast oxidative glycolytic muscle cells. And there's a fast glycolytic. And like usual, they give you a hint in the name. These are all muscle cells. They're all moving. They're all contracting. Turns out this slide lists them in order of speed. And they tell you the name. Slow oxidative fibers are the slowest of all 3. They contract the slowest. Fast oxidative glycolytic fibers are kind of in the middle in terms of the speed of their contractions. And fast glycolytic fibers are the fastest of all. Why do their speeds vary? It has to deal with how they use ATP. And to use ATP, your muscle cells actually use an enzyme. No, you don't need to know it. But how much energy you use will actually affect how fast you could contract. So if you use a lot of energy quickly, you will contract quickly. While if you use or burn through your energy kind of slowly, you will move a lot slower. So your slow oxidative fibers are the slowest because they use energy kind of slowly. They have what we call low myosin ATPase activity. That's the enzyme your myosin heads are using to hydrolyze ATP. They have very low myosin ATPase activity. They're going to burn through ATP slowly, so they're going to move slower. While fast oxidative glycolytic fibers have kind of a little higher myosin ATPase activity. They use ATP a little faster, so they'll be able to move faster. And fast glycolytic fibers have the highest myosin ATPase activity, so they're going to burn through ATP very quickly, and they'll be able to move faster. But there's a consequence. Turns out the faster you burn through ATP, well, the faster you'll get tired and hit a fatigue. So it turns out those slow oxidative fibers, because they're moving or burning through ATP so slowly, it's almost like they're going to have a lot of ATP for a long time. So these slow oxidative fibers could have a long, sustained contraction. So where do you tend to find these cells? Where do you tend to find the slow oxidative fibers? Let me give you a hint. Think of muscles that don't usually move that fast. It's going to be in your back. It's your back muscles again. Your back muscles don't really move that quickly, not like your fingers or your eyelid muscles. They move very rapidly. Your muscle cells in your back don't move that fast. But your back muscles could hold a contraction for a long time. Remember, you're sitting up or standing up because of your muscles in your back. And you could sit up or stand up pretty much all day. Why? Because you have lots of slow oxidative fibers in the muscles of your back. They could sustain a contraction. Your fast oxidative glycolytic fibers, remember, they're a little faster or they have a little bit more myosin ATPase activity, so they're going to burn through the ATP a little bit faster. Think about the muscles in your upper limbs. Some of them have some fast oxidative glycolytic fibers, so they can move a bit faster. But again, because they move a bit faster, they're going to burn through energy a little bit faster and fatigue faster. That's why you can't lift and drop your arms up and down all day, every day very quickly, because they'll get tired after a while. And then there's your fast glycolytic fibers. They're the fastest because they have the highest myosin ATPase activity, so they're going to burn through the ATP rapidly. So these cells are going to get tired the fastest. Where do you find these fast glycolytic fibers? You find them in places like your fingers. Your fingers can move extremely quickly because they have these fast glycolytic fibers. They're burning through ATP quite quickly. That's why you could take notes kind of fast. But if you were to try to write really fast with your fingers, after a while, you'll probably get a hand cramp. Why? Because you've run out of your energy. You've hit that muscle fatigue and cramped up. That's why you can't really write that fast for that long, because you're going to burn through the ATP, because these are fast glycolytic fiber cells. So now you know, not only do you have different types of muscle tissues where the cells look different, within a single muscle tissue, the cells could also look different. And then to finish off this chapter, we just kind of talk about some changes we'll see in skeletal muscle and how you could change it. And then finally compare it to the other two types of tissue, cardiac and smooth. So next thing I mentioned is how can we change skeletal muscle tissue? Well, there's two major ways to do it. We're talking about training. We're talking about working out. So think there are two major ways to work out, at least according to anatomy and skeletal muscles. There's something called endurance training and something called strength training. To help you kind of wrap your mind around it, think of endurance training as aerobics and think of strength training as lifting weights. Okay. So what's some changes we're going to see in endurance training? Think marathon runners, people that run and build up endurance are doing endurance training. This is when you do frequent contractions and you're not focusing on weights. Okay. This is basically running. When you're running, you're doing frequent contractions in the muscles of your legs, but you're not necessarily trying to lift something heavy. You're running. You're focusing on frequency, not the weight of the load. That's endurance training. And when you focus on frequency of contractions, when you do endurance training, you're going to see some changes. Turns out your muscle cells are going to become more efficient at breaking down sources of energy and using them. Your muscle cells literally get better at making energy. This is why when you notice some people, especially when they work out a lot and do a lot of aerobics, it seems as if they could eat more food than you and not gain weight. Well, that's not quite true. They can eat more food, and it does seem like they're not gaining weight. It's not anything special they are doing. It's because their muscle cells are getting more efficient at using their sources of energy. So you'll see that change in cells during endurance training. They become more efficient at using energy. What about strength training? Oh, what about lifting weights? Oh, you see something happen when you lift weights. Think of bodybuilders. They lift weights all day. What happens to them? They get bigger. With strength training, you will see muscle cells literally get bigger. That's what happens when you lift weights. When you lift weights, your muscles don't get bigger because you get more cells. That does not happen. So if you were to look at someone like Ronnie Coleman or Arnold Schwarzenegger back in the day when they were big guys and compare them to a skinnier individual, those guys didn't have any more muscle cells than the smaller person. It's just that their cells literally got bigger. And we give a name to that. When an organ gets bigger, because the cells making it up get bigger, we call that hypertrophy. 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 That's when an organ gets bigger because the cells making it up gets bigger. And that's what happens to your skeletal muscle during strength training. Your skeletal muscle cells hypertrophy. They get bigger. This is different from something else called hyperplasia. Hyperplasia, Hyperplasia. Hyperplasia is another way an organ could get big. But in hyperplasia, the organ gets big because it increases the number of cells making it up. This is something that occurs in something called BPH. Stands for benign prostatic hyperplasia. It actually told you in the name. This is something that happens in males because males have a prostate and females don't. This is what you see usually in older males. It's simply their prostates get bigger. Spoiler alert, when you get older as a male, your prostate is going to grow. Why? Because the prostate undergoes hyperplasia. The number of cells making it up actually increases when a male gets older. But again, we're sticking with muscles, skeletal muscle cells. When you work out, doing strength training, think lifting weights, you're going to see hypertrophy. The muscles are going to get bigger because the cells are getting bigger. And that's pretty much skeletal muscle. And remember, there are other tissues. There's cardiac and smooth. I'll mention them, but like always, stick to the exam review. Now that we know basic anatomy of muscle cells, thanks to skeletal muscle, we can finally compare it to cardiac and smooth. And next up is cardiac. And like usual, you know things already. And we're going to take some of this skeletal muscle tissue information to help explain cardiac. Don't worry, it'll make sense as we go through it. Just first up, we have to remind ourselves about cardiac muscle tissue. Remember, when you look at cardiac muscle tissue under the microscope, you're going to see those alternating light and dark bands. It has striations. But now you know what really is a striation. You know striations are alternating I and A bands within a sarcomere. So just by noticing that cardiac muscle tissue has striations, you automatically know that if you were to look inside the cells, you're going to find sarcomeres made up of alternating A and I bands, and you're going to find myosin and actin and troponin and tropomyosin. You're going to find your typical muscle cell anatomy, or a very similar muscle cell anatomy, at least when it comes to the striations. But there are other things. Remember, you do not control cardiac muscle tissue, so you don't really have to worry about troponin and tropomyosin the way you do with skeletal muscle tissue. And you remember, another major difference is that cardiac muscle cells typically have one nucleus per cell, while we saw that in skeletal muscle cells, they had multiple peripheral nuclei. So that's it. Remember, stick to the exam review. I'm just kind of glossing over cardiac and smooth muscle tissue. Then next up is smooth muscle tissue. Again, we already know it's called smooth because it does not have striations. So now you know something. Because it does not have striations, you know it does not have alternating A and I bands. So it does not have sarcomeres the way that cardiac and skeletal muscle tissue does. So how is this one able to contract? Well, it could still contract because it still has those contractile proteins. And that's what you see on this picture. It still has the thick and thin filaments. It's just that they're not arranged in that unique pattern that we see within a sarcomere. They're kind of scattered in these oblique ranges that you can see in the picture off to the right on their segment C. The blue, thicker looking bands are the thick filaments, and the yellow looking bands are the thin ones. So it still has actin and myosin. It's just not in that pretty arrangement of alternating light and dark areas because there are no sarcomeres. But because it still has the protein, you could still do contractions within smooth muscle. Also, remember, you don't control smooth muscle, so again, you're not concerned with troponin and tropomyosin to that extent. Because there are no sarcomeres, there are also some other structures missing from the smooth muscle. For example, you won't see an extensive sarcoplasmic reticulum the way you did in skeletal muscle, because remember, the sarcoplasmic reticulum was there to store calcium, which you needed to move troponin and tropomyosin out the way. So if you're involuntary, you're not really worried about troponin and tropomyosin. So you're not as worried about having calcium. So you're not going to have it stored in a sarcoplasmic reticulum. So you won't really see that in smooth, and you really won't really see that big of a sarcoplasmic reticulum in cardiac muscle either. So that was just a little brief comparison of skeletal muscle tissue to cardiac and smooth. And again, these last couple slides are just us finishing off muscle tissue. Again, this slide is just us mentioning those definitions we talked about again. When your muscle tissue grows, or when your muscle organ, appears to grow, aka when you're working out as an adult, it's getting bigger via hypertrophy. Why? It's because the cells get bigger. You don't increase in number. Remember, increasing in number is hyperplasia. And muscle cells, skeletal muscle cells, don't increase in number. Why? Because they don't really undergo mitosis once you hit adulthood. Remember, mitosis is when cells kind of go through the process of separating DNA and eventually splitting into daughter cells to make more cells. That doesn't occur in adult skeletal muscle tissue. That's why you don't see hyperplasia in skeletal muscle. You don't grow more muscle cells once you hit adulthood. They just simply could either get bigger or smaller based off whether or not you work out. So again, hyperplasia is increasing in size due to increase the number of cells making it up. And hypertrophy is when you get bigger because the cells get bigger as well. Don't worry about smooth muscle and pericytes. And then the last two things we talk about are where do your muscle tissues come from and what happens as you get older. And when it comes to development, just think back to tissues. We know this from tissues. Remember, at one point, you were just made up of 3 major layers. There was endoderm, ectoderm, and mesoderm. Remember, muscle tissue came from mesoderm. And I just want you to know that again. Don't worry about any of this other development of muscle tissue. Just know, or muscles in general. Just know muscle tissue originated from mesoderm, which was just an embryonic type of tissue, which is a tissue before you're born. And then lastly, we talk about what happens when you get older. You know by now it's not good. Things go down the drain. Even your muscles do. How so? Turns out as you get older, you're going to damage muscle tissue. And when you damage it, it's likely to fibrose. Why? Because you're not likely to replace it with other cells. Why? Because you don't do mitosis. Remember, you're going to do hypertrophy, not hyperplasia. So once you damage muscle tissue, it's likely to fibrose and get replaced with connective tissue, get replaced with collagen, And this is going to happen steadily throughout your life. And by the time you're in your elder years, possibly 70s, up to about 40% of your muscle tissue can be replaced with this type of scar tissue. And this helps to explain stuff. Because you're replacing muscle, which is for movement, with connective tissue, where you're not going to have that much movement. So you're going to see some changes in muscles, the organs, as you get older. What? Where you're going to notice that the muscle strength will decrease. Why? Because there are not that many muscle cells anymore, because they've scarred and become